Good afternoon, all the participants and our distinguished speaker for today's event, Dr. Vikas Jadon. And I'm getting this opportunity to introduce him. And welcome, sir. Welcome to Lovely Profession University. And if I briefly explain, because he's having a long list of achievements, because it will took me more than 15 minutes if I'm going to read his biodata. So briefly, I'm going to introduce Dr. Jadon. He is Associate Professor at Department of Biochemistry and Biotechnology, Sardar Bhagwan Singh University, Dehradun. If I look into the experience, overall experience, academic as well as his research experience, he's a very diverse kind of a researcher as well as a faculty member, because he is having a teaching and research experience more than 16 years in the area of that biotechnology and biochemistry. And during his tenure, these 16 years of tenure, he has put his hands into the different fields of biotechnology and applied sciences. He's basically a person who is having a rich experience in plant molecular biology, environmental biotechnology, and if I talk about that environmental biotechnology, especially waste management, because this is the area where we have to give a certain attention as a citizen of India, as well as just our significant contribution into the society. Because this is a major challenge for today's world to manage the waste in terms of that biological waste in terms of those electronic waste, because that is going to cover that earth. And they are going to cause certain diseases. They are going to pollute the environment. They are going to pollute that air, water. So we have to find certain innovative solutions. By that we can minimize that waste, which is a household waste, which is the waste from the industry, especially those textile industries, distilleries, and the industries like pharma industries, because they are just heavily working on those antibiotics, and they are putting their effluent in the rivers and the nearby those crops, and people they are living in that area, they are getting affected with them. So I'm just coming back to that my speaker's experience, because he has delivered so many lectures as a resource person on the themes related to that environmental biotechnology. He has conducted so many funded workshops for government agencies. He has guided students on wastewater or environmental biotechnology research. And he has published book chapters, research papers, and he has presented so many papers in that conferences or other scientific platforms like conferences, symposia, and if I go for their different area, he's also having his research experience in plant molecular biology, bacterial interactions, and he had put some foundations in terms of that research publications or whatever those gene sequences submitted in NCBI. And he is actively involved in that postgraduate research also. And if I go for that, his citations, H-index, again, he's having more than 330 citations. He's having H-index 10 or more than 10. He's having I-10 index more than 10. And if I go for that, his PhD tenure, because that's quite important, he has done PhD on a topic that is really my prime area of interest, that biochemical characterization of eucalyptus F1 hybrids through isoenzyme analysis. So, and he has done so many awards also. And if I look for his administrative role, academic role, he is having significant contribution in terms of research as well as academic leader. So I invite Dr. Jadon uh, to start his talk for today's guest lecture. Welcome, sir. Please, I request you if you can start your lecture.
Thank you, sir. Thank you so much, Dr. Umesh. And am I audible?
Yes, sir, you are
Yes, sir.
audible.
Yes, sir. Yes, sir. You are audible, sir.
Oh, okay. Thank you. Thank you so much for inviting me for this webinar. And thank you, Dr. Umesh, especially for such an elaborate introduction for me. And thank you, Dr. Vikas Sharma, and all the students present here. Today I see a diverse, uh, participation from students of various faculty, and basically majority I hope will be from biotechnology itself. And as Dr. Umesh has said about me, that I have diverse uh, uh, research fields, right? So basically that depends upon the student's interest. Whosoever is interested in either conducting more in environmental biotechnology, so I assign a topic to him or her on the this topic only, or core interest. My core interest has always been the plant and microbial interaction. And today, because it's a very important aspect, that we are living in a polluted world now, and most important part of a life is water. So any thing related to uh, pollution in water is a serious concern for the society, for mankind, and it's a prime importance, and it's a key role for a biotechnologist, basically, to combat such challenges and take such challenges, and uh, be involved in innovations that could finally lead us to the for combating such challenges of environmental pollution. So I'll just share my screen to you all. Just give me a second. Yes. So my screen is visible to you all.
Yes, sir. Yes, sir. It is visible. It is visible.
Okay, so without taking further time, I'll directly come to my topic, that is wastewater treatment. And today I'll be covering this wastewater treatment in two aspects. First will be, because I can see the students from first year also, and obviously from masters, there will be the participation from master students as well. So a very basic aspects of water treatment technologies will be my first part, and what all physical and biological methods are used for the treatment of wastewater. And further, I'll be elaborating few of my past researches, what we have done at Sardar Bhagwan Singh University in the Department of Biochemistry and Biotechnology, and currently what our core group of researchers we are planning to do or we are doing. Many of the aspects we have right now in the pilot scale, and many of the projects are in the pipeline to be funded by the Central Department of Science and Technology, New Delhi. So uh, I'll be talking about wastewater treatment, and When we talk about wastewater is, you know, everybody knows it. It's 99% of the pure water, and there are certain uh, foreign things which are added to it, which either change the texture of the water, or it changes the odor of the water, or it changes the physical or chemical attributes of the water. And when it is in the negative side, we term it as a sewage. So sewage is nothing but a water which carries waste, right? And the prime importance will be to separate out these waste from water and recycle it back, because we have a limited source of fresh water. Everybody knows this, and the prime concern globally is to face the water crisis in the coming years. So it will be very prudent that we develop technologies that could recycle the water, because conventional methodologies are there, but we need to improve more. We need to have more elaborate strains. We need to have more less hazardous biological techniques which could combat to the treatment of this wastewater. So when we characterize this sewage, we characterize into two parts. That is the domestic wastewater and the industrial wastewater. So domestic wastewater will be the complex matrix which contains the proteins, which contain organics, which contains chemicals, basically from the community discharges, right? And the TDS ranges from 350 to 1200 mg per liter, and the COD, chemical oxygen demand, it ranges from 250 to 1000 mg per liter. And the most concern is, of course, because it's the community discharge, though, the most concern is the pathogens, the microbial load, and that is up to 109 numbers per ml. And then, of course, there are many nutrients in form of lipids, in form of proteins, in form of other organic waste, and heavy metals and micropollutants. The prime concern is, of course, the industrial wastewater. And industrial wastewater contains most of the hazardous chemicals and heavy metals, which includes lead, mercury, cadmium, or the industrial wastewater, which is nearby the ores or the fossil fuel power plants. They have another type of hazardous materials with radioactive compounds and many other things. So we need to have technologies that could treat these wastewater, right? And that water which is treated can be recycled back, and it could be used for community services, either for drinking purpose, of course, and if not, for irrigation or other social amenities. So we need this treated wastewater, of course, to prevent the groundwater pollution. This is the most important part, because if we collect this discharge either from the community discharge or from the industrial discharge, we have to prevent the seepage into the groundwater, because the local body which resides nearby the community or nearby the industry, whether it is a pharma industry, it's a coal mine, or it's a power plant, so the groundwater contamination or the pollution is one of the most important aspect which has to be addressed. Then another is to prevent the seashore pollution, of course, to prevent soil pollution, because most of the areas near the wetlands, uh, because wetlands are reducing and the land which is left behind is a contaminated soil. So we need to prevent, or we need to reclaim that soil also. So one of the aspect will be to prevent the soil pollution. And once this groundwater or the fresh river basins they are polluted, they will, of course, they will go and pollute the marine life. So that is one of the prime concern nowadays. And of course, on top of all, the protection of the public health, right? Then another aspect of why we need to treat water is to reuse it, because again I'm saying we have a limited resources and limited resources of fresh water. So our prime concern is to reuse the treated effluent, right? Either for drinking, and if not, for agriculture, right? For groundwater recharging. And many of the industries, they have their own devices for recycling of industrial wastewater for their own industrial use, and of course, by solving the social problem caused by accumulation of wastewater. Because wherever the wastewater or the standing water will be there will be n number of other nuisance which will be created by these wastewater. So these are the basic aspects why there is a concern to treat the wastewater. So the technical goals, if we see, of course, when we are saying it's 99% pure water and impurities are suspended in it. So first and the prime technical goal will be the separation of solids from liquid, right? Then the stabilization of the separated solids, because these are the pollutants, right? They cannot be discharged anywhere, but they have to be stabilized before they are discharged in any of the system. Then, when we are concerned about the community discharge, the disinfection of the pathogenic microorganism will be the prime concern, because otherwise it will pollute the local river bodies, and finally it will lead to the hazardous community infections. So that has to be addressed, right? And finally, the proper reuse of disposal of treated liquid and solids, right? So the big concept that we will be discussing will be the wastewater treatment process. So first I'll be dealing with the conventional, what we learn in our books, what we should know about the treatment process, because nowadays when you go around for amusement parks, for community centers or big firms, they have their own system. Like many of you must be visiting Hyderabad, and you are very aware of Ramoji Film City, right? So Ramoji Film City, as you enter into it, you will see the very big tanks of the aeration tanks, the aerobic treatment of wastewater. So they have the complete system, and they what they use, what they discharge, they recycle back, and they use it for drinking purpose. So see what efficacy they are utilizing, the resources they are utilizing, that they convert their wastewater to the level of drinking water, right? So the big concern will be the wastewater treatment process, right? And it will be, uh, subjected into three parts. First is a primary treatment, then is a secondary treatment, and then is a tertiary treatment. So primary treatment will be the removal of any solid or any grit, right? So basically these are the settling tanks, and after settlement of all the heavy particles, all the heavy solids, then this water is subjected to the secondary treatment. And in secondary treatment, we have aerobic and anaerobic lagoons, or the bioreactors. I'll be dealing with some latest technologies which are employed over the conventional one. Then it has trickling filters and sludge oxidation ditches, and mostly they're employed to reduce the biological oxygen demand. And I hope when we are in this webinar, so most of the students, they are well aware of the terms BUD, biological oxygen demand, and chemical oxygen demand. So I'm not going into the depth of these concepts, but general overview. Then after this reduction, around 80% of reduction in BUD, finally the treated water is subjected to disinfection. And this majorly involves the chemical treatment, which involves the, if the nitrate level is above the permissible level, the nitrate removal will be there, the phosphorus removal will be there, and finally, if it is subjected to the drinking purpose, it will be disinfected, right? Then if you come to first domestic water, wastewater, the characteristics includes, this is the complete chart of what the approximately BOD levels are there, because the more densely crowded community, the higher will be the BOD and COD level. and if the community level or the population density is less, of course the domestic wastewater will be a weak. It will be categorized as a weak system with lower BOD rates, right? So these are the limits of BOD, COD, organic nitrates, then the total nitrogen, phosphorus, and total solids and suspended solids, right? So in first part, when we talk about primary treatment, the total solids removal is basically subjective. So when we talk about the industrial wastewater characteristics, so these are basically, because that is basically from the industries, and the prime concern will be the chemical attributes of the wastewater system. And in this we have pH that ranges from 6 to 10, so more alkaline pH. Temperature is around 40 degrees. Suspended solids is 400 mg per ml. So this is the total profile in which we get the toxic metals also. We get cadmium, cyanide, sulfates, oil, grease, and many other things which are micropollutants which are suspended in this industrial wastewater. So when we talk about the primary treatment, so primary treatment is often called as clarification, right? Clarification, which is subjected to settling and sedimentation of the uh, heavier particles. If you can see all this heavier particles, they all settle down, and this sludge is separated out, right? So when we talk about the settling tanks, the capacity and the retention time are different, right? We talk in terms of retention time, how much time the wastewater is retained in, allowed to settle. It is retained in the tank. So approximately around 2 hours or 3 hours, the first untreated waste water is subjected, and it is allowed to settle down. And then the clarified liquid effluent is further subjected to, the secondary treatment process. So the benefits, of course, is reduction in the suspended solids, which otherwise will cause many hindrances, because when we are talking about RBCs and trickling filters, so heavier grit or heavier particles will ultimately choke the system, right? So it's most essential that the first primary settling of this wastewater should be done. So maximum time, a percentage of BOD is reduced in the primary settling, and reduction in the amount of wastewater sludge in the activated sludge plant is further subjected. Okay, and of course the removal of floating materials and all these things, they are done in the primary treatment. Then it is subjected to the secondary treatment. And as you can see in this figure, this is the huge tank, aeration tank, in which the aerobic treatment is being done, right? So the secondary treatment basically is the biological treatment plant part. So it is biological wastewater treatment, in which we employ microorganisms like bacteria and fungi to remove the pollutants from wastewater through biochemical reactions, right? So, biological, when we classify this biological waste methods, we classify into two parts. One, which is the criteria which is based on the suspended and the attached treatment process, right? So in suspended growth process, the biological material, or biological, I refer to microorganisms which are subjected to the treatment process. So in suspended growth process, the biological microorganisms are maintained in a suspended form. So they are in a homogeneous form. If you see the previous slide, if we talk about this tank, so this tank will, you can see a heavy aeration in this, and you can see the froth. So in this, the microorganism is completely in the suspended form, right? But as we talk about the trickling filters in the next few slides, we'll be talking about trickling filters, in which we have a concept of biofilms. So when we talk about films, that means the microbes, they are attached or adhered to a source, right? So when we are classifying these methods, so one way of classifying is suspended growth processes, and second will be the attached growth process. But when we talk in terms of metabolism of microorganism, then we classify this whole process into aerobic and anaerobic system. So aerobic treatment system will be in the presence of molecular oxygen, and anaerobic will be, of course, in the absence of molecular oxygen. And the concept is very clear. You must have gone through the biochemical processes, the basic biochemistry, where the final electron acceptor is oxygen. So that will be aerobic. And if not, any other inorganic component is a final quencher, then it will be the anaerobic part. So these are the broad classifications that are used when we talk about the wastewater methods, right? So the previous slide, that was the activated sludge system, and it is a aerobic suspended growth biological wastewater treatment methods, right? In which activated sludge means the microorganisms, they adhere and they grow on the suspended solids, right? When you आप जब लस्सी भी बनाते हैं, उसे आप बहुत अच्छे से churn करते हैं, तो उसमें flocks बनते हैं. Similarly, when we are talking about activated sludge system, so aeration causes the formation of flocks, right? And flocks will be nothing but the organic substances which are suspended in the water system, and over that becomes the adheration part for the growth of microorganisms. And that colony, a small colony, a microsphere, that is called as a activated sludge. As you can see in this previous slide, this part, all the froth and all the suspended flocks will be the activated sludge. And activation we are saying because activation means the growth of microorganism on that particular flock. Otherwise it is dead, right? So we talk in term of MLSS, mixed liquor suspended solids. So that will be the mixture of wastewater with bacteria in the aeration tank. And when we talk about the kinetics of aeration tanks, MLSS will be one of the major part that we'll be discussing. So this is the principle of activated sludge. Here Q will be the wastewater flow rate, and SO will be the concentration of pollutants such as BOD. So to the wastewater flow rate along with the BOD will be subjected to the aeration tank. And in aeration tank, X will be the mixed liquor suspended solid concentration that we were talking about, the MLSS. Because MLSS will finally define the load of the aeration tank. You can just you cannot just fill up the entire tank and choke it. So there should be an optimum concentration, and every aeration tank you have to optimize the MLSS concentration with bacterium concentration, right? Then S will be the concentration of dissolved pollutants in the aeration tank and the effluent, and V will be the volume of the aerated tank. So aeration tank will be the volume, final volume of the aeration tank, and what MLSS you have poured in, along with the concentration of the pollutants, because it will be the pollutants that will be oxidized and that will be reduced to the inert component, right? Then it will be subjected to secondary sedimentation tank, where the activated sludge is settled. If you stop churning, the whole sludge will settle at the bottom, and this will be. Hello, yes, any queries? Okay.
No, sir, there are no queries. Please mute. I think it is by mistake. Some participant has unmuted.
Okay. No problem.
Okay, sir.
So Then it is subjected to secondary settling tank with the seed. This activated sludge, it is used as a seed material for recycling back to the aeration tank, where the concentration and the growth can be maintained. So this is the whole principle, where the flow rate, the waste sludge flow rate, the return sludge flow rate, it is all maintained, and finally the aeration tank is the functioning of this aeration tank is maintained. So second aspect, this aeration tank was of course the suspended system, and the stabilized or the attached growth biological system will be the trickling filters. I'll just show you the picture. You can see here. This is the aerial view. This is the top view. Please, I request to mute your mics, please.
Yeah, Abushad, please mute yourself. Abushad, please. Yes, thank you. Please, I request all the participants to remain muted. And after the end of the session, you can have your questions. Okay. Thank you.
Right. Thank you. So you can see this aerial view of trickling filters, and these are the sparges which are spraying the wastewater over the matrix. This is the matrix, and if you can see the cross section, so this is the wall or the matrix on which the wastewater is sprinkled over, and these are the fans that rotate. This will be the rotation, right? And while rotating, they are sprinkling, or very homogeneously they are sprinkling this water over this trickling filters. And why we are calling it as a trickling filter, because the water that is sprayed over this matrix, it trickles down, right? It slowly trickles down, and while it trickles down, this matrix is a very uneven surface. So all this suspended solid, what we are calling it as MLSS, that will be adhered on this wall. It attaches to this wall, this whole wall, and finally over those attached MLSS, there will be the growth of microorganisms, right? And that growth of microorganism further will lead to the formation of an attached biofilm, right? And attached biofilm will trap all the pollutants, all the organic load, all the inorganic load, and finally what will be collected in bottom in the reservoir will be a treated water. So it will be examined, and if again recycling is required, again a second phase of recycling will be done over this trickling filters. So basically this contains a tank, a rotatory distributor, a filter media. A filter media will can be of crushed stones or gravel or plastics or even fibers, right? And finally, the under drain system, which will collect the whole this treated water. And beneath this system, the bottom of this whole trickling filters are the ventilators, right? So they are the aerators which sprays air from the bottom, so that the anaerobic environment is being constantly created while the treatment process is going on. So this is the attached growth biological system, this one. When we talk about anaerobic system, because trickling filter and activated sludge, rotating biological contractors, lagoons, these are all the examples of conventional aerobic treatments, right? And when we talk about the efficiency, when we are related to the efficiency of the wastewater treatment, then we come to anaerobic treatment, because it is one of the most efficient system which do not require the molecular oxygen, and finally it treats the high solids such as animal manure, biological sludge, night soil to the digested form. So this is of course the biological process that is carried out in the absence of molecular oxygen for stabilizing the organic materials. And finally, the end product will be the conversion into either methane, that is a biogas, carbon dioxide, ammonia, and finally the biomass. So, since we are saying that there is no external electron acceptor, the product generated is the breakdown of organic matter, and the final quencher of ETS will be, of course, any inorganic substance. Like if we talk about anaerobic fermentation of glucose, right? So glucose will turn into energy or pyruvate, or electrons will be generated, and this pyruvate will be directly converted anaerobically into ethanol, right? Otherwise, if you go through the aerobic process, the byproduct will be something different. Similarly, there are so many other end products, that is the production of gases like H2S, biogas, that is methane, then nitrogen, carbon dioxide, and all these are the anaerobic respiration of glucose. And it depends upon the preference of the electron acceptors, and finally the end product, it changes. And I hope this whole thing you must have gone when you are studying biochemistry, basic biochemistry. Okay, so finally, okay, please mute your mics, please. So if you see the basic microbiology that is involved in this anaerobic uh, system of digestion. So we have prominently four classes. One is the fermentative bacteria, and where the biotechnology comes is in the strain improvement, or the bioprospecting of such anaerobic forms, which could lead to the efficient removal of the BOD and COD levels in the fermentation process, right? So like we are also engaged in SVS, we are also engaged in research. One of our faculty and basically one of the group is involved in the bioprospecting of such microorganisms which can anaerobically degrade many of the pollutants which are present in the wastewater. So here where the role of biotechnologists come, because these are the conventional forms which are known to all. Clostridium and Lactobacilli, Bifidobacterium, these are the common forms, but the consortiums, the combinations of the microbes which can be used, either the bacterium consortium or the fungal consortium, which can be used for the process of anaerobic bacteria, is one of the main aspect on which we are working. So first group is fermentative bacteria, then second group which are hydrogen producing bacteria or acetogenic bacteria. So the end product, basically on the end product, these are categorized. Then acetogens are there, which are involved, like Clostridium and Acetobacterium. And finally, the broad group which is involved in anaerobic digestion are the methanogens. Right, so methanogens, they have microbes classified as archaea and the primitive bacteria, right? And two main classes which metabolize acetate to methane are methanoaceta and methanosarcina, right? And further researches are more for bioprospecting of more methanogens are going on for the use in this system. So when we talk about anaerobic part, like aerobic, maybe talked about trickling filters or activated sludge process. The anaerobic system is basically, it depends upon the capacity of a bioreactor that you're working on, because it's a closed system. You have to maintain the anaerobic environment.
Hello? Hello, yes, sir. Sir Vikas, sir, your voice is not audible.
Yes, hello? Hello?
Hello? Yes, sir. Sir,
Sir,
voice is not audible. I think there is some problem in the
just a second. I will call him.
इतनी देर लगाओगे क्या?
Hmm? Yeah, hello, sir. Unmute yourself. Unmute yourself, sir.
Okay.
Yeah,
Sorry, there was a network issue.
no problem, sir. No problem.
Okay, so I'll resume back. Just a second.
Okay, sir.
Okay, so my screen is visible now?
Yes, sir. Yes, sir. It is visible. It is visible. Now you can continue, sir. Thank you.
Okay, thank you so much. Okay, so I'll be just winding up because time is a constraint. So this figure shows, the process, which are the types of anaerobic reactors which are involved. And previously, the conventional parts are the anaerobic ponds, septic tanks, which we use in the household process, and Imhoff tanks, which have a loading capacity of 1 to 2 kg COD per meter cube per day, right? So this is a lower capacity and much more advanced versions of these anaerobic reactors. They are now available, which includes your upflow anaerobic sludge blankets and fluidized bed reactors and anaerobic sequencing batch reactors, which have a loading capacity just more than 2 or 3 times than the conventional part. So I'll just wind up with what we are doing here at SBS University, Dehradun. Basically, one of our core research area was on the decolorization of the textile dyes, because in here in Uttarakhand we have many textile industries, and from here to the UP and Kanpur and all these areas, they are rich in textile industries, and they are involved in coloring the textile fabrics, and finally all these textile dyes, which are azo dyes, they are discharged into the water streams, and this causes the decolorization of water. The water, it turns more turbid, it becomes black, or it becomes colored, so that the intensity of the sunlight, it is minimized, and finally the aquatic life, or the biological life is affected. So what we have done is we have isolated many of the bacterial strains, which we have given to the state pollution board here in Uttarakhand also, and it is in the trial stage, where we have used Bacillus, like one of this species which I am showing here is the Bacillus strain, and this strain is capable of decolorizing the dye, azo dye. So you can see this is the color of the dye, and in 12 hours and in 24 hours, the color is totally uh, degraded, decolorized. right? Because it breaks the ring structure, and there are key enzymes which are involved in the breaking of these ring structures. In SO dyes and many other dyes also, we have conducted many of the experiments with few microorganisms which are capable of decolorizing these dyes within 24 hours. And we have submitted these strains to the State Pollution Board, and along with the Uttarakhand Council of Science and Technology, we are in a in a way of making the uh, reactors which will contain these microbial forms for decolorizing of such wastewater. Then in this research only, we have localized many enzymes which are involved in the decolorization of the azo dyes and many other dyes. And in xanthomonas, we have mostly done in xanthomonas, and we have kinetic characterization we have done for azo reductase enzyme, which is a key enzyme for reduction of this color. Then, of course, we have purified azo reductase because we were in the uh, process of making some immobilized enzyme system which could directly be used for decolorization of the wastewater. Then we have identified, or we are involved in bioprospecting and screening of more fungal and bacterial strains. We have very good fungal strains which are capable of decolorizing these recalcitrant dyes. Otherwise, these dyes they impose, because they are recalcitrant, they are not degraded by the sunlight or any other chemical process. Otherwise, if you treat it with heavy chemicals, then that chemical treatment itself will be a threat to the water system. That's why we want a convenient system. As I said earlier in the beginning of my presentation, that we need a safe system for the treatment of water, right? Otherwise, we have chemicals, we can treat directly, we can treat the water with chemicals, but the chemical that will be used for treating that particular wastewater itself will impose hazards. So using these microbial forms, uh, this is a very convenient system and a safe system, because biological system, if you just heat up the system, or you just add basic chemicals, these will be degraded off, and we can get rid of all this system, and the pure water can be separated off. So for the wastewater treatment which are contaminated with the dyes, we have provided many strains. Then the upcoming project that we have submitted to Central Department of Science and Technology Water Technology Initiative, that is a development of smart nanobiosensors for rapid detection of most common pathogens in water resources. Because now as there is a overload of public, and the domestic discharges are more, and these domestic discharges, they are directly vented into the river system, into the local river system, and community infections, they are transmitted from a person through the domestic discharge to the river system to another person. So this becomes a route. So a basic thing is that there should be a rapid detection method for common pathogens. So what strategy we have proposed for combating this situation is that we have a pathogen. We all are students of biotechnology. So we need to know that there is one antigen and there is one antibody, right? So what we propose is that we will take an antigen. Antigen will be the part of a pathogen. Say if it is E. coli, so it will be a part of its cell wall, or it will be any other antigen, or it will be a complete E. coli, right? So we have already raised antibodies against few antigens, pathogens that are contaminating the local water system. And these antibodies, after purification, will be immobilized on the nanobars. That will be a nano system which are immobilized over a suitable uh, matrix, and that will be connected to a sensor. So as soon as the antibodies which are immobilized on the nanobars, it will interact with the pathogen that is present in the water, because this system will lead to the identification of a very low titer of pathogens. Because the major limitations when we talk about the detection of pathogens in the water system, that we need to culture it, and if the concentration of pathogens is very low, then we get a false results. So maybe when we are able to make these nanobiosensors, so this will be a ready system where we have immobilized the antibodies, specific antibodies against a specific pathogen. So that will be immobilized over these nanobars, and the signal will be transduced, and the detection will be there that yes, there is a presence of pathogen in this water system or not. So this will be a qualitative detection, and finally, whatever pathogen we have identified can be treated accordingly. So the first major thing is how you detect the presence of a pathogen. So this is one, and we propose to have two strategies in this. One is the solid state nanopore based nanobiosensors, and second will be the liquid junction membrane based nanobiosensors. So this is in a process. Part of antibody production we have already completed, and I hope we'll be soon will be coming with a product. Then so you can see this is the basic concept of nanobar, over which the antibodies they are attached, and they will react with the antigen or the pathogen, and it will immobilize this pathogen. So this will be the nanobar with antibody and antigen immobilized, and the signal will be transduced, and that can be sensed. So this is one. And the second indirect method will be, because we have pathogens, we know the microbial pathogens, they are infested with phages. There are n number of phages which are specific for many of these pathogens. So we have developed many PCR-based screening methods in which we screen phages, the detection of phage. So if there is a detection of phages, we can indirectly correlate it with the detection of the pathogen present in the water sources. So we have developed many PCR-based screening methods, and we have synthesized many degenerative primers for the identification of these phages, and soon we will be publishing our results in the journals. Another aspect on which, because my keen area is plant biotechnology, so we keep on doing experiments on various things. So what next we have in line is the development of cell cultures from hydrophytes. So hydrophytes, we won't be growing the hydrophytes in the morphogenic cultures, but we will be developing callus. We'll be deriving the callus from these hydrophytes, and finally these callus, because this is a clump of cell, right? These are the clump of cells. So we have methods by which we can actually disintegrate this whole cluster into a cell form. So this will be the cell cultures. So these cell cultures, we are subjecting it to various pollutants, and we are identifying, we have identified 2 or 3 pollutants which are absorbed or which are being structurally modified by these cell cultures. And because we are targeting many wetlands in Uttarakhand and in Uttar Pradesh, so these contaminated wetlands will be screened out, and these hydrophytes will be introduced, which are capable of either degrading. See, there are 3 things: either the degradation, or the structural modification, or adsorption or absorption. Right, these 4 things, every hydrophyte or every cell respond in a different manner with the pollutant that is involved. So this is the upcoming research that we are into it, and soon we'll be developing a protocol for identification of such things under in vitro conditions, and finally it will be upgraded to uh, in vivo testing. So these are our collaborators. We are having funds from and projects from Uttarakhand Council of Science and Technology. Then we are the collaborators of with the State Pollution Control Board, because in many water technology initiatives, we are with the pollution board. And then we have scientists from IIT Roorkee, which are engaged in the characterization and the designing of bioreactors for Abito. We are working on a pilot scale, and then we will be upgrading the system. And of course, the State Water Testing Lab in Dehradun. Major instrumentation is done there. So I hope you all will be benefited from this. And for all the students, I'll just say, that use your innovative minds and come up with innovations, because any simple idea can click, and it can create wonders. So thank you all the organizers for inviting me for this webinar, and thank you for patient hearing. Thank you.
Thank you very much, sir. Thanks a lot for such a nice lecture. Actually, if I'm going to brief your lecture, that starting from that basics and what research you are going on, that you have inculcated in your presentation, and you have shown your collaborator also, and as well as maybe we were listening your presentation, and it gives that idea that where we can pick up those threads, that we have to apply our mind to find out the problem in our surroundings, and what kind of knowledge we have, so which tool we can use. So especially I get benefited with that. I honestly give my views for that, and maybe I assure you that the student or the faculty member, they are connected with us from last 1 hour. They also got good ideas from your presentation, and I'm highly obliged that you spared your time, and you have shared your thought process as well as your ideas in between that very short period of time, because it's not easy while you are working from last so many years, and you have to summarize your work, or you have to connect with student, faculty, and within 1 hour you are going to complete your, but you were so targeted, and you have finished your talk within timeline, and thank you very much for that. And I hope that whenever this pandemic will over, and we'll get a chance to invite you in our university, and we'll have one-to-one interaction with you. And
my pleasure.